There is a really great diagram in your textbook. This is on page 339 that is going to exceed all of my abilities at drawing anything for you guys regarding transcription and translation right now. So I want to walk you through this diagram. I think that once I do that, it can help you kind of really grasp what is happening in this entire process. So the first thing I'm going to do is I'm going to block off this entire section here. This section is all translation. And based on the first video that you just watched, transcription comes first in protein synthesis. So I want you to take stock of what you see here. First, you have the DNA, that's this double helix part. You have this section where this DNA has been unzipped. Now, they're telling you about RNA polymerase, and that's not an enzyme that I'm particularly worried about you guys knowing about. Ultimately, it's really just making the RNA strand. But I do want you to know that there is an enzyme that has to unzip this strand of DNA first, and that enzyme was helicase. It actually unzips and unwinds the DNA. So once it does that, then this RNA polymerase can come in and start transcribing one side of this DNA. Now, what happens during transcription is that the DNA sequence is used to make an RNA sequence. Okay, but keep in mind there are a couple of differences between DNA and RNA. So if you have a DNA sequence of A, T, G, G, C, C, A, T, T, and I just made that up, so don't go trying to find that anywhere. But if you had this DNA sequence, and you know for a fact that it must be DNA because there are T's, and thiamine is only found in DNA, then when you are transcribing it to RNA, so if this is DNA, we are transcribing it to RNA, we have to think, okay, what does adenine bond with? Now, normally adenine will bond with thiamine, but remember, there is no thiamine in RNA. And in fact, thiamine is replaced with uracil. So A bonds with U. Thiamine always bonds with adenine, no matter what. Okay, so if you have thiamine in DNA, you've got adenine in RNA. The guanine and cytosine pair does not change, so that should be the easiest part because it doesn't change. The same as with DNA, guanine pairs with cytosine, cytosine pairs with guanine. Here you have adenine again in DNA. Remember, adenine pairs with thiamine usually. However, in RNA, thiamine is replaced with uracil, so adenine pairs with uracil, and then thiamine pairs with adenine. So then you have an RNA sequence, and what I want you to notice here is that every 3 letters of this RNA sequence is a codon. Okay, now that word is very important because a codon, a single codon, codes for one amino acid. And we'll look at when that happens. That's actually going to happen in translation. But you need to go ahead and know that every 3 letters is effectively a code for an amino acid. Okay. So let's go back to our diagram here and look at this RNA chain that is being made. Now this is going to be the messenger RNA. If it's the messenger RNA, it's providing a message for someone. So what's going to happen is after this RNA chain has been made, and some things actually have to happen to it, it has to be processed, but again, that's more detailed than we really need to go into. But once that RNA chain is ready to leave the nucleus, it does, and it leaves the nucleus through these nuclear pores, and it travels to a ribosome. Now, a ribosome is made up of 2 parts, and that's what it's showing you here. You've got the small subunit and the large subunit. And there are these 3 sites in a ribosome. And I love this diagram because it's really showing you what is happening in each of these sites. Okay? So the A site, and the way you can think about this is this this RNA strand is being read like a tape. So it's as if this ribosome is moving along the RNA strand. Okay, so it first read these three letters, this ACG. And remember, that is a codon. And what's happening, remember we said that the tRNA, transfer RNA, is a RNA molecule that brings amino acids. So here's this tRNA molecule. There's the amino acid that is attached to it. And it has an anticodon. This anticodon is going to be complementary to the codon on the mRNA, so that this tRNA knows, hey, it's time for me to bring my amino acid here. So when you have ACG on the codon, you'll have UGC on the anticodon. And the codon is actually what you read to figure out what amino acid is going to be brought to the ribosome. So as the ribosome continues reading the RNA strand, another tRNA molecule comes in. So ultimately what is happening, and I know I'm doing a terrible job of explaining this, but I will show you guys a video in class that I think will help you again. But this, for example, this tRNA molecule has brought in a specific amino acid. This tRNA molecule is bringing in another amino acid. This tRNA molecule over here is going to bring the next amino acid. And what happens as this ribosome reads the tape is these amino acids are going to get attached to each other. And that's the job of the ribosomal RNA. These amino acids are going to get bonded. And remember that the bond in between amino acids is a peptide bond. That's why this is called a polypeptide. So as the ribosome continues down the length of the mRNA and continues reading the strand, more tRNA molecules are going to come in. They're going to deposit their amino acid. The amino acid will get attached, and this strand will continue to grow. And then the tRNA molecule will leave. Okay, so watch the video that I'm about to show. But first, let's look at how a codon codes for an amino acid. And this is also in your textbook on the opposite page. This right here is called a coding dictionary. And I'm going to show you this one. I'm going to show you how to use this one and how to use another one that I think is a little bit easier, but there's no telling which one they'll give you on the EOC. So what happens is every 3 letters codes for an amino acid. Well, this table, this coding dictionary, is organized so that you can follow the sequence of letters and figure out what amino acid it's being coded. So back to our sequence that we just made. Our first sequence was UAC. Okay, so that means that our first letter being U, so we're looking at the first base here, it's U, so it's going to be somewhere in this location. Our second base was A, so now we know it's going to be somewhere here. And our third letter was C, so we're going to go across and find UAC, that codes for tyrosine. Okay, so really you're finding where all 3 of those letters intersect. U, A, and C intersect right here at tyrosine. Our next one was CGG. C, G, and G is right here. Arginine, codes for arginine. Okay, again, you go to the first letter is C, second one is G, so you're looking at where those meet up, and then the third letter is G as well, so you've got CGG. The third codon was UAA. So we can start at U, our second letter was A, and our third letter was A, and that is a stop codon. A stop codon is not coding for an amino acid. It's only signaling to the ribosome to stop translation, and then it's done. All right? So after this, watch the video on transcription and translation. Watch it several times if it helps. I think that if you talk your way through it, and we'll watch it in class as well, talk your way through it, it'll help you understand the concept much better.